Imagine treating a toddler's birthday party like you're navigating an active minefield.
Oh, yeah. It's incredibly stressful.
Right. For the parents of the roughly 2% of children in Western countries with a peanut allergy, that is just the baseline reality.
Yeah, that's their everyday.
Strict avoidance isn't just a clinical recommendation. It is this psychologically exhausting lifestyle of interrogating waiters and policing play dates and treating a generic bakery cookie as a lethal threat.
It really takes a toll.
It does. And today, we aren't looking at a way to simply manage that fear. We are going into a deep dive on a stack of research showing how a daily skin patch can systematically hack a toddler's immune system to fundamentally unlearn the allergy.
Right, which is a massive shift.
Yeah, reducing the real-world risk of an accidental reaction by nearly 80%.
The therapeutic landscape for pediatric food allergies is it's undergoing a massive shift right now. And the focal point of that shift is epicutaneous immunotherapy.
Okay, so epicutaneous meaning through the skin. Exactly. Through the skin. And we are synthesizing 3 pivotal pieces of data today regarding the VP250 VIA skin patch.
Which delivers 250 micrograms of peanut protein through the skin, right?
Micrograms, yes. And we have the phase 3 EPITOPE trial subgroup analysis, which was published by Skurlock and colleagues in 2025. We also have Benjamin Remington's 2019 Monte Carlo risk reduction simulation.
Which is fascinating, by the way. I can't wait to get into that.
Yeah, it really contextualizes the risk. And finally, we're looking at the long-term compounding data from the EPOPEX open-label extension study led by Dr. Greenhot.
Okay, so the core EPITOPE results already set a pretty high baseline. In toddlers age 1 to 3, 67% of those wearing the VP250 patch for a year responded to the treatment.
Right.
Compared to just 33.5% on the placebo.
And that's a huge delta. Right, but comparing those macro-level trial results to the chaotic biology of an actual toddler is where this gets really interesting. Because taking a pill for an allergy is sort of like trying to rewrite your computer's operating system by dumping random code into a Word document.
That's a good way to look at it, yeah.
It goes through the stomach, it enters the bloodstream, and it just triggers systemic chaos. But VP250 is fundamentally different.
It's highly targeted.
Exactly. It's like downloading a software update directly to the immune system through a localized port. You apply the patch, and it quietly reprograms the immune cells in the outer layers of the skin without ever dumping the allergen into the bloodstream.
Right. And the basic EPITOPE results are impressive, but the real story is hidden in the subgroup data.
Mm-hmm.
Because the mechanism driving that software update relies entirely on Langerhans.
Langerhans cells, okay.
Yeah, they reside in the stratum corneum, which is the outermost layer of your skin. The patch creates this condensation chamber on the skin.
Oh, like a little greenhouse effect.
Essentially, yes. It solubilizes the peanut protein, so it can penetrate just those outermost epidermal layers.
Just the very top.
Exactly.
Huh.
And because the epidermis is non-vascularized, meaning there are no blood vessels there, the antigen doesn't reach the mast cells in the bloodstream.
Which is what prevents the whole body anaphylaxis.
Precisely. That prevents systemic anaphylaxis. Instead, the Langerhans cells capture the protein right there in the skin and migrate directly to the regional lymph nodes.
They carry away like a little messenger.
Yeah, like couriers. And there, they interact with naive T cells and preferentially push them to become regulatory T cells.
Or TREGs, right?
TREGs, yes, which actively suppress the allergic Th2 response.
Okay, which brings us right to the core of Skurlock's 2025 subgroup analysis. Because clinical trials notoriously prefer pristine patients, right? They want people in a vacuum to isolate variables.
Always. It makes the math easier.
Right. But toddlers with peanut allergies are rarely pristine biological canvases.
Not at all.
They usually present with an entire cluster of overlapping immune issues driven by what we call the atopic march.
Right. And Skurlock's analysis is vital because it tests the efficacy of this localized Langerhans pathway against the systemic noise of a highly distracted immune system.
Because in reality, kids with peanut allergies usually have a cluster of other issues.
Exactly. And the baseline demographics of the 362 toddlers randomized in the EPITOPE trial really reflect that messy clinical reality.
I have the numbers here. It's wild. 80.1% of them presented with concurrent atopic dermatitis.
Eczema.
Yeah, eczema. And 66.9% had concomitant food allergy.
Which is the CFA acronym you'll see in the data.
Right, meaning their immune systems were actively fighting off dairy or egg or tree nuts on top of the peanut allergy. And then 18.2% suffered from asthma.
So these are complex kids.
Okay, let's unpack this for a second. If my kid's immune system is already distracted fighting off dairy, egg, and asthma triggers, wouldn't it be too overwhelmed to properly process a peanut patch?
You think so?
Yeah, I mean, if the system is actively flaring, fighting off an egg exposure, managing severe eczema, the systemic Th2 inflammatory response is operating at a 10.
Right, the alarms are blaring everywhere.
Exactly. It feels highly counterintuitive that the immune system would have the biological bandwidth to just calmly accept and process a tolerance-building signal from a tiny patch on their back. I'd expect the treatment efficacy to just plummet in those subgroups.
That is the logical assumption, assuming the immune system functions as a single, easily exhausted engine.
But it doesn't.
No. The data proves it is highly compartmentalized. The statistical analysis showed VP250 was superior to the placebo across every single one of those subgroups.
Really?
Yeah, there was no significant interaction effect.
So it worked just as well.
What's fascinating here is how the presence of ongoing asthma or eczema or other food allergies did not blunt the immune system's ability to learn from the patch.
That is wild.
The responder rates hovered tightly between 64 and 72% for the VP250 cohort, regardless of whether the child was simultaneously managing asthma or multiple other food allergies.
So the software update still installs perfectly, even if the computer's running 10 other heavy background applications.
That's exactly it, because the local skin-to-lymph node circuit that's utilized by epicutaneous delivery remains completely isolated from the systemic Th2-driven inflammation circulating in the blood.
Oh, okay.
The Langerhans cells are still efficiently capturing the antigen and trafficking it to the lymph nodes to induce those regulatory T cells, and they're completely unaffected by the systemic immune battles happening elsewhere in the body.
So they have their own dedicated highway.
Essentially, yes.
Okay, so that isolated circuit explains the efficacy, but applying a highly concentrated allergen to a child with severe atopic dermatitis raises a massive red flag on the safety side for me.
That's the natural next question.
Because if a child's skin barrier is already compromised, weeping or inflamed from eczema, introducing an epicutaneous peanut patch every single day for a year seems like throwing gasoline on an inflammatory fire.
And that was a major concern. Scurlock's team tracked the SCORAD metrics with intense scrutiny for that exact reason.
SCORAD is the standardized scoring for atopic dermatitis severity, right?
Yes. They monitored it very closely, and the data revealed absolutely no clinically meaningful change in overall eczema severity from baseline to month 12 for the children wearing the active VP250 patch.
Wow, so it didn't make their eczema worse overall.
No, it didn't.
I mean, the local application site reactions were present, obviously.
Yes, of course.
You're inducing a local immune response, so mild to moderate erythema or pruritus where the patch sits is to be expected.
Right, the redness and itching.
But it didn't trigger systemic eczema flares across the rest of the body.
Exactly. Most adverse events, the Ts, as they're called in the study, were overwhelmingly confined to the application site. They were mild to moderate.
Okay, but, however, evaluating the severe systemic safety endpoints, specifically anaphylaxis, requires looking at the full architecture of the trial data, not just the active treatment arm.
Right. You have to look at the placebo group to really understand the context.
Because I'm looking at the safety tables from Scurlock's analysis right now, and there's a glaring anomaly to me.
What stands out to you?
Well, the rates of anaphylaxis were significantly higher in the children who had atopic dermatitis or concomitant food allergies and were receiving the VP250 patch compared to the kids on the active patch who did not have those conditions.
Right.
It's like 7.7% versus 4.0%. So my first instinct, reading that, is to blame the patch for exacerbating severe reactions in highly allergic kids.
And that is exactly what a lot of people think when they first glance at that chart. But look at the control group columns for those exact same demographics.
Okay, let me trace this across.
You see it.
Yeah, the kids wearing the dummy patch, the ones receiving absolutely zero peanut protein, experienced the exact same elevated anaphylaxis rates if they had eczema or other food allergies.
Exactly. If we connect this to the bigger picture, these higher rates of anaphylaxis were also observed in the placebo group.
That is such a vital distinction.
It really is. The increased risk of severe reactions is an inherent function of their baseline biology. Toddlers presenting with severe eczema and multiple food allergies simply possess a more highly reactive immune phenotype.
They're just more sensitive in general.
Right. Having eczema or multiple food allergies inherently makes a child more reactive. They are more prone to anaphylactic events in their daily lives, regardless of the intervention.
Right, they're reacting to other things in their environment.
Precisely. So the trial data successfully captured the natural volatility of the disease, proving the elevated severe reaction rate is not a side effect of the VP250 patch itself.
Oh, okay. I see. It's like noticing that people who carry umbrellas tend to get struck by lightning more often and blaming the umbrella.
That's a perfect way to look at it.
When you really look at the control group, you realize just that people with umbrellas are out in the storm.
Yes.
And the underlying highly allergic biology is the storm here.
Exactly. The umbrella cannot be blamed for the lightning.
That makes total sense.
We do, however, need to factor in the asthma subgroup data, which presented a slight but distinct deviation in patient retention.
Right, because while respiratory events like cough and wheezing were higher in the kids with asthma, they were again balanced with the placebo group.
Right. The baseline disease activity was the driver there, too.
But the actual treatment difference showed up in the discontinuation rates, didn't it?
Yes, it did. There was a slightly higher permanent discontinuation rate for asthma patients. It was 7.7% for kids with asthma on the VP250 patch compared to just 2.4% for the non-asthma group.
Oh, okay. So why is that? If it's not causing more anaphylaxis, why are the asthma kids dropping out more?
Well, while the patch didn't drive higher rates of anaphylaxis in the asthma cohort, the daily burden of managing minor respiratory side effects alongside the treatment protocol was just enough to tip the scales for a small percentage of families.
Ah, I see. It's just too much to juggle.
Exactly. It is a critical nuance for clinicians who are counseling parents. The safety profile holds up robustly across the atopic march, but the daily management threshold might be slightly lower for families who are already exhausted from managing pediatric asthma.
Right. Every family has their breaking point with how much they can manage on a daily basis.
Precisely.
Well, moving from those kind of abstract clinical trial endpoints like eliciting doses to the practical reality of a family's daily life, what does this medical data actually mean when a toddler reaches for a snack?
Right, because the clinic is very different from the real world.
Totally. I mean, a 300 milligram eliciting dose during a double-blind, placebo-controlled food challenge is a great metric for the FDA. But it means absolutely nothing to a parent who's staring at a box of generic cookies in the grocery store.
No, it doesn't help them at all in that moment.
Which brings us to Remington-Eyre's 2019 quantitative risk assessment, which I think shifts the perspective entirely.
It does. Remington-Eyre's team bypassed the standard clinical threshold reporting and utilized what are called Monte Carlo simulations to model the chaotic variables of human consumption.
Okay, for anyone listening who isn't familiar with Monte Carlo simulations, it's essentially using computational algorithms to predict the probability of different outcomes when there are just too many random variables to calculate normally, right?
Exactly. They ran 5 million modeled eating occasions.
5 million.
5 million. And they specifically targeted 4 categories of packaged foods that are notorious for accidental cross-contamination and high recall rates.
Which are cookies, donuts, ice cream, and salty snacks.
The absolute staples of a toddler's diet.
Right, exactly. The stuff that's at every single birthday party. And to build an accurate Monte Carlo model for this, you can't just simulate generic eating. You have to feed the algorithm probabilistic distributions for every single variable.
And they were incredibly thorough with this.
They were. They took retail surveys across Europe and North America to map the parts per million concentration of peanut protein that's typically found in contaminated batches of these specific foods.
Right, they're the real-world manufacturing data.
And then they crossed that contamination data with anthropometric data detailing the exact portion sizes a 1 to 3-year-old toddler actually consumes in a sitting.
Because a toddler doesn't eat a whole box of cookies, they eat maybe 1 or 2.
Right. And they layered those consumption and contamination variables against the dose distribution curve of patient thresholds gathered from the EPITOPE trial.
They effectively built a virtual environment populated by the exact immunologic profiles of the trial participants.
And then they just fed them virtual contaminated snacks 5 million times to calculate the precise likelihood of an unexpected allergic reaction.
Yes. And the baseline calculation for an untreated peanut allergic child is staggering.
It really is.
The algorithm estimates an 11.3 to 29.7% chance of an unexpected allergic reaction over a single year, solely from consuming those 4 types of packaged foods.
Just from cookies, donuts, ice cream, and salty snacks.
Just from those. On the high end, that is nearly a 1 in 3 chance every single year that a toddler will suffer a reaction despite strict parental avoidance.
That is terrifying. That probability distribution perfectly quantifies that psychological exhaustion we discussed at the very beginning of the deep dive.
Yes. Strict avoidance is a mathematical gamble heavily weighted against the patient.
But here's where it gets really interesting. When Remington's model applied the immunity thresholds of the children who had been on the VP250 patch for 12 months, the probability landscape inverted.
The drop is dramatic.
The yearly risk of a reaction plummeted to between 3.4 and 5.9%.
Which equates to a massive 73.2 to 78.4% relative risk reduction for the treated population.
Over 70% risk reduction just from wearing the patch for a year.
Right. And to ensure the integrity of the model, they ran the placebo group through the exact same simulation.
And what happened to them?
The children on the dummy patch experienced a relative risk reduction between negative 0.3 and negative 3.9%.
Oh, wow. So virtually zero improvement.
Statistically zero improvement, yes.
This specific data point completely redefines the utility of epicutaneous immunotherapy for me. Because this patch isn't a magic wand that lets a kid sit down and eat a whole peanut butter and jelly sandwich.
No, definitely not. That is not the goal of this therapy.
It is an internal airbag. It is explicitly designed to protect you from the invisible, accidental cross-contamination crashes that happen at birthday parties and in grocery store aisles.
The airbag analogy is perfect. Achieving a bite-proof clinical status fundamentally alters the chronic management of the disease.
Bite-proof, I like that term.
Yeah, it's a common term in the field now. A 78% relative risk reduction transitions the allergy from a state of constant, acute, life-threatening vulnerability to a manageable condition.
It gives parents a chance to breathe.
It does. It builds substantial safety margins directly into the child's immune system. The psychological relief that provides to parents is just massive. It alters the daily anxiety of navigating a world that is just filled with processed foods.
So we've established that the airbag works after 12 months, but the obvious mechanistic question is durability.
Right, does it last?
Exactly. Does this protection degrade over time? Do the kids build a tolerance to the treatment, or does the treatment build tolerance in the kids?
It's a vital distinction.
Like, does the immune system eventually just adapt to the patch and say, oh, I'm used to this signal, now I'm going to stop responding?
Answering that requires analyzing the third pillar of this deep dive, which is the EPOPEX study.
This is the open-label extension of EPITOPE, right?
Yes. Dr. Matthew Greenhot and his team published this long-term extension data in 2025. And the most revealing data comes from a very specific subgroup, the crossover subgroup.
Okay, the ones who were on the placebo first.
Exactly. These were the children who spent the first year of the original trial on the placebo patch, which established a solid, naturally progressing baseline for them. And then they received the active VP250 patch for 3 consecutive years.
So having a full year of placebo data for that specific cohort proves that their allergy wasn't just spontaneously resolving on its own.
Exactly. We know they were truly allergic and remaining allergic.
And when they finally started the active treatment, after 1 year, 62.7% reached an eliciting dose of 1,000 milligrams or more during a clinical food challenge.
Which is a significant amount of peanut protein.
Right, let's put that in context. A typical peanut kernel is roughly 250 to 300 milligrams of protein. So 1,000 milligrams equates to tolerating 3 or 4 whole peanuts before triggering a reaction.
And again, this is a child who previously might have reacted to a fraction of a milligram in a contaminated cookie.
It's incredible. But the compounding effects observed in years 2 and 3 are where the mechanism of action truly reveals its depth.
Yes. By the end of year 3 on the patch, the percentage of children tolerating that 1,000 milligram threshold jumped to 71.2%.
From 62% in year 1.
Right, and more profoundly, 46% of that crossover cohort reached an eliciting dose of 2,000 milligrams or more by year 3.
So nearly half of these toddlers could consume the equivalent of 6 to 8 whole peanuts without reacting.
Yes.
And they achieved this simply from wearing a skin patch containing a fraction of a single peanut's worth of protein. It is this highly leveraged biological feedback loop.
This raises an important question about how the immune system is actually adapting over time, because this proves the body is undergoing a fundamental compounding change.
It's not just a temporary fix.
No, the continuous exposure is not exhausting the immune system. It is actively and continually reinforcing the regulatory T-cell response. The desensitization pathway is compounding. It's learning to ignore the allergen more effectively with each passing year.
Which we can actually see reflected in the safety trajectory over that 36-month period, right?
We can. The severity of the reactions during the clinical food challenges steadily decreased over time.
Right. I think at 12 months, 19.2% of the children who did react experienced severe symptoms. But by 36 months, that dropped to just 10%.
And even the local application site reactions, so the redness and itching on the skin, those continued to decrease over time as the epidermis adapted to the daily condensation chamber.
So their skin just got used to having the patch on?
Basically, yes. But most critically, looking at the long-term safety data, there was 0 treatment-related anaphylaxis in the 3rd year.
0?
0. The longer the antigen is delivered via this isolated Langerhans circuit, the safer the patient becomes, and the higher their biological tolerance climbs.
So the immune system isn't just temporarily exhausted or temporarily pausing its Th2 response. It is actively undergoing these durable epigenetic modifications to just permanently ignore the allergen.
Exactly. It's a true biological re-education.
Wow. Okay, let's recap this journey because we have covered a massive amount of ground today.
We have.
We've tracked this from the murky diagnostic reality of a toddler's immune system right through the mechanics of epicutaneous delivery. Scurlock's analysis proved that the VP250 patch cuts through the noise of the atopic march.
Right. It maintains its efficacy and its systemic safety, even if the child is battling severe eczema or asthma or multiple food allergies.
And then Remington Armis Monte Carlo simulations proves that this localized cellular re-education mathematically slashes the real-world risk of packaged food reactions by over 70%.
It provides that crucial internal airbag.
Yes. And finally, the EPOPEX data confirmed that this internal airbag only gets stronger and safer the longer it is used.
It really is a paradigm shift. We are witnessing a fundamental move from strictly avoiding an environmental threat to actively engineering systemic tolerance through the skin.
So what does this all mean?
Well, if we pull back and look at the broader implications, it introduces a pretty provocative concept.
Already.
Think about it. If a simple low-dose epicutaneous patch can successfully and safely hack a highly reactive toddler's immune system to unlearn a deadly allergy over 3 years,
Right.
could this continuous passive skin delivery model eventually be used prophylactically in infancy?
Oh, wow. Before they even develop the allergy.
Exactly. The infant immune system is remarkably plastic, and their epidermal networks are incredibly active.
Right. They're still figuring out what's dangerous and what isn't.
Precisely. So could we one day use temporary skin patches on healthy newborns to program their immune systems to accept all major food allergens before an allergy even has the chance to develop?
Just bypassing the atopic march entirely?
Bypassing it entirely,
Right.
treating the allergy before it ever exists.
That is an incredible thought to leave on. Thank you so much for joining me to break this all down.
It was my pleasure.